Good morning, good afternoon, and good evening. Thank you for your interest in today's webinar by the IQ Green Chemistry Working Group. My name is Frank Roschanger. I'm from Boehringer Ingelheim, will be your moderator, as mentioned. It's my great pleasure to introduce our two speakers, Sharon Michalak from Amgen and Mike Kopach from Eli Lilly. Our first speaker, Sharon, is originally from Niagara, Ontario, Canada, where she completed her Bachelor of Science in Chemistry. She then became tired of the Canadian winters and moved to Southern California to complete her PhD at UC Irvine in 2019, with her research focusing on natural product synthesis. Sharon decided to stay in California and joined Amgen in Thousand Oaks as a process development scientist in the pivotal and commercial synthetics group. She currently leads the Green Chemistry program at Amgen Process Development, and she's an active member of the IQ Green Chemistry Working Group. Our second speaker, Mike, earned his PhD in organic chemistry from the University of Virginia in 1995 with Professor Harmon. He then did a postdoc in natural product synthesis at Colorado State University with Professor Myers. Mike began his industrial career in 1997 at Roche, where he focused on development and implementation of commercial processes for several drugs, including the first synthetic peptide produced on ton scale. In 2001, Mike joined Eli Lilly, where he spent 14 years working with small molecule APIs, including Olumiant, that launched in 2017, and is presently in use as a COVID-19 therapeutic. Afterwards, Mike helped build the synthetic peptide development division at Lilly, where he is now an executive director and was responsible for the launch of tirzepatide. Additionally, Mike is quite active leading efforts to advance sustainability in the synthetic peptide field. Over the past decade, Mike has represented Eli Lilly not only at the IQ Green Chemistry Working Group, but also at the ACS Green Chemistry Institute Pharma Roundtable, including 2 tenures there as a co-chair. With this, I'm happy to turn it over to Sharon and Mike so they can tell us their exciting story on a metric for a greener future in drug manufacturing, iGAL 2.0.
All right, thanks, Frank. Thanks, Frank, for an outstanding introduction. We really appreciate that, and this is a topic that we're certainly really excited to deliver our progress in this very important area. Next slide, please. Okay, so what we're going to talk about today for this webinar is primarily about pharmaceutical industry sustainability, and really how can companies, and not only companies, but even like academic institutions, others that are practitioners of green chemistry, how do we, very importantly, best measure sustainability? That's kind of a key feature to take away today. What are the best metrics to use? How do you use them? And you'll see that we've developed some very important tools. So you'll see these different tools or these different green chemistry metrics throughout the presentation, and we'll explain them in more detail as we go. But a real important foundation is the process mass intensity and the E factor. That's the basis for some of the more sophisticated metrics we'll talk about, such as the innovation green aspiration level version 2. And what we will do today, very importantly, is Sharon will give us later on a live demonstration of the active tool we have that's available for use. Next slide, please. So what specifically is our IQ Green Chemistry Working Group? Well, we're certainly part of the IQ Consortium, and we're really one spoke on the wheel, on the hub, so to speak. The IQ Consortium is composed of numerous different working groups, and one of which we are. And really what this group is a collection of companies coming together to work pre-competitively. And so the whole idea there is to deliver transformational solutions to our industry that not only benefit us directly, but also patients, regulators, and the broader R&D community. And the real benefit here is we just get much more traction with our message when it comes from many companies working together as one voice, which in this particular sub-team, we have 13 companies, rather than any individual company kind of going it alone. And so there's been great success stories in the IQ Consortium. And we have, I believe, one to add here with the progress we've made on this green chemistry working group. We're very much focused on helping influence regulatory policy, educating regulators, but it's beyond that. It's also providing tools and educating on sustainable chemistry and engineering. We've made a lot of progress in that field over the last 5 years. We have some key papers that we've published more recently. The iGAL 2.0 metric, which we'll talk about quite a bit today. And we did give an introductory presentation on this topic by Stefan Koenig and Aaron O'Brien a couple years, and we do have a refresh here. This metric we will talk about. We're really excited about the group and how it's expanded. And you'll note here on the Sustainable Chemistry and Engineering paper that we're targeting United Nations Sustainable Development Goal 12, which is focused on sustainable consumption and production of API. And in that paper, you might notice on the bottom right corner, we've included Roger Sheldon has made great contributions to this work. And for those of you who don't know, Roger Sheldon is the original inventor for E-factor. I believe it first published, if my memory serves, back in around 1992, so about 30 years ago. So absolutely great to have him on board with all our companies that are trying to steer our oars in the right direction of more sustainable R&D processes. Next slide, please. So what is green chemistry? That's in some way is really the toughest slide, because there's a very subjective part to this, because you ask any number of people this question, they'll come up with different answers. I like the one, I'd say green chemistry is good chemistry, excellent chemistry, the best chemistry, striving towards greener. Those are important things. We do show here the 12 principles of green chemistry. Credit where credit is due. These principles were established by Anastas and Werner nearly 25 years ago, and really provided pharma and many others some key principles for us to anchor our sustainability strategies too. I'm not going to go over all these individually, other than to say that these principles are linked, and certainly one key one is number one, prevent waste. If anything, remember the prevent waste principle, because if we do a good job with that, a lot of the other principles sort of come along. It's along the lines of, right, if we lose some weight, like, which I need to do, I need to reduce my E factor or process mass intensity, then other things happen that are good, such that, you know, your cholesterol and all those things go down. Same kind of relationship with the 12 principles of green chemistry. And we've had Nobel laureates that have weighed in on this, and others. This is the sustainable direction that we are going as an industry and society. Next slide, please. So what are the benefits of sustainable manufacturing? And certainly, this is where it has real teeth. The pharmaceutical and generic industries produce greater than 100 billion kilograms of API per year. And our best estimate is the average E factor, or total waste, is about 150 kg of waste per kg of API, which that translates to greater than 15 billion kgs of co-produced waste, and an annual cost of about $30 billion. I will comment that this information was generated some years ago, so we expect that this is a great underestimation, that the actual annual waste disposal costs are probably much higher. So there are some real economic teeth to addressing the waste issue. And for those that are new to the pharmaceutical industry, we show a graphic below that describes the life cycle for a pharmaceutical API. Where you start is all the way over on the left, where someone in your drug discovery organization or collection of scientists will discover the next, discover a candidate that'll have some interest for a clinical indication, then that will actually ultimately progress through different levels of clinical trials. And while those clinical trials progress, and we're going left to right here, that's where significant development does occur in the pharmaceutical industry. So you'll have to have actually active pharmaceutical ingredient campaigns to specifically support the clinical trials, and those give great opportunities to install green chemistry sustainable type processes. And so really where green chemistry applies is throughout the entire development life cycle, numerous opportunities. Obviously, when you get to the end, where you launch a product and you're fortunate enough to have a drug that's going to submit to the FDA, that's where it becomes very important to have your green and sustainable process for scale-up manufacturing. Next slide, please. So we'll go through this slide pretty quickly, but there's really at present no one metric to describe all the sustainability attributes. And the couple key foundational metrics we do have, which are closely related, are complete E factor and process mass intensity. And what these metrics are the total kilograms of all materials that go into a synthesis divided by that are required to produce 1 kilogram of API. The only difference from difference PMI has from complete E factor is complete E factor is PMI minus 1. Next slide, please. And again, the complete E factor metric is based off of the original metric, as mentioned earlier, developed by Roger Sheldon back in 1992. That original E factor metric did not include water. Complete E factor does now include the water metric, which is very important. Key point here is our ideal E factor is going to be 0. Again, that's what we strive to obtain. So if you think about how could you do that, would be if you had basically chemical reactions that operated completely neat and generated no waste. Next slide. So complete E factor certainly measures core production waste in the API life cycle. So this is a slide that shows the key part of what a pharma industry will do is we focus on the core production. Those are the synthetic steps in our API manufacturing process. You'll note, though, however, there are two key parts to this that are outside of core production. One is upstream, and the upstream component is basically all the things that go into produce all the components that go into raw material production, and that does have a sustainability component. On the other side, and it's what the patient sees is the packaged product or the device, and there certainly is a large sustainability component to that as well as for distribution of your product to get it on the shelf. And so, but the key area of focus here is the core production area. That's the area we have direct control of, and it's our API synthetic manufacturing process. Next slide, please. And so why should we measure API manufacturing waste? Well, a key reason to do it is certainly that we can determine the positive impact over the development life cycle on our development, and certainly take credit for all the improvements that we make as an industry. And so we did benchmark this in our 2021 manuscript, looking at the complete E factor of early development processes, late and commercial. Late would be ballpark phase 2, phase 3. Early would be phase 1 and everything before, and commercial would be for our launch products. You note here the trend is very positive, that we have E factors as high as 600 for our early process. We cut that in half by late development, and then get a further reduction down to 100, an E factor of 182 for an average commercial process. On the right's a very important graph where we can estimate the contribution of outsourced manufacturing to the complete E factor. And you see the metrics are roughly about 50% today. For the mid stage of development, it's right, or for the late development, it's actually 60%, and a little bit under 50% for commercial manufacturing. And so what we'll say for that is that the pharma industry has changed over the last 2 or 3 decades. If we did the same type of survey 20 or 30 years ago, 95% of all the work would be insourced into most pharmaceutical companies and very little done externally. Most of the pharma companies now have evolved towards global entities, where in fact we're utilizing key technologies wherever they occur in the world, and so that doesn't mean that these processes that are external to our companies don't have oversight. They in fact do have oversight, but by using our partners outside of our brick and mortar buildings, that gives us great opportunity to even further improve the sustainability credentials of our processes. Next slide, please. And so there are some good tools out there in the public domain to use for calculating process mass intensity, again, which is the kg of all materials that go in to produce 1 kg of API. On the bottom left, there's a PMI calculator that's provided by the ACS Pharmaceutical Roundtable that's on their website. Just go there and anybody can download it and use it. It's very straightforward. Bristol Myers Squibb has done an excellent job on developing a PMI prediction tool by basically looking at their entire portfolio and even looking outside of that to come up with PMI targets for individual steps. And so, for example, they can go as far as determine for an individual reaction what an average PMI value should be for that reaction. So if they, for example, have a process that runs 2 or 3 green yards, they'll say, okay, this is the typical PMI for a green yard type process. And so that's a very, very powerful tool. Also on the right is a smart PMI tool for Merck that looks at, that evaluates, that's based off of complexity. And that's a specific tool that Merck uses. Next slide, please. And so key piece here is that the PMI complete E factor certainly does not capture the full picture. We're certainly focused here on core production a piece because that's what we have in direct control and what we can measure. However, a very critical piece here is measuring the waste for starting materials that go in and having standards for those starting materials. Next. And certainly, it'd be great to have the ability to be able to benchmark your process, your synthetic route, with not only what you're doing in your own company, but also benchmarking it to other pharmaceutical processes, basically. And we believe we have a very good way to do that and to also capture process improvements. Okay, let's go next. Okay, let's go next. And so a critical thing here is that all API are not created equal. And I think any one of us who've worked in this field or have worked on different academic targets will realize that we can get a very significant difference in complexity going from one material to another, particularly in small molecule. And that's shown here for 2 examples. One is dabigatran and the other is clenbuterol. Very, very different materials in terms of complexity. And so certainly for the more complex material like dabigatran, which has a fairly high molecular weight of 628, you see that that's broken down into some simpler components, but also have some complexity. And so a key question is, how do you capture the contributions from a sustainability perspective for those smaller components? And so a key aspect here is to be able to establish a smart API waste target per asset with the understanding that you know, all assets are not created equal. Next, please.
Okay, so I will take it over from here. Thanks, Mike. That was a really great introduction on green metrics and PMI and E-factor and really the scope of what we're looking at here. So I have the opportunity to present the Innovation Green Aspiration Level 2.0. So the iGAL original publication came out in 2018, which the title page is shown here on the left. iGAL was introduced as a way to compare a broad range of small molecule targets that we face in the pharmaceutical industry and set waste goals that keep our industry accountable to our impacts on the environment as we continue to treat unmet healthcare needs. The metric was created from a large data set from the IQ and the ACS Green Chemistry Institute member companies, and it allows you to measure how green your process is relative to industry standard data and created as a unifying green chemistry metric that allows chemists and engineers to measure the innovation impact they have on the pipeline programs. Because in my opinion, at the heart of green chemistry is innovation. And hopefully by the end of this webinar, we can have conveyed how iGAL 2.0 can provide a way for scientists to effectively communicate their added innovation value during process R&D. And you can see this is, I'm giving you a quick little preview to the scorecard here, which I will go into a little bit more detail on for all of these quadrants. So from user feedback, we found that we ended up updating the metric to include more intuitive sustainability indicators, yield and convergence, which I'll cover in some later slides. But overall, these new indicators enhance the fit of the statistical model. And I also want to showcase that iGAL is a smart metric, one which addresses some of the issues Mike had alluded to earlier, where traditional PMI and E-factor alone may not suffice. So a metric that is specific, measurable, attainable, relevant, and time-based. So before I go into the metric, I have to preface the iGAL rule. A very important part of developing iGAL was establishing a consistent starting point for what we define as raw materials. Admittedly, when I first started learning about E-factor, it wasn't necessarily clear to me on where do we start our counting. In a world of completely integrated life cycle analysis on our pipeline programs, we would be able to define our starting materials back to earth, wind, and fire. But this level of analysis will differ between companies, and we need to define a common starting point. And that is $100 per mole from the Sigma Aldrich catalog or other common commercial suppliers, but no bulk or specialty quotes. So the goal here is that we are trying to bring any molecule, regardless of how complex it appears, back to the same building blocks, ones that are less than $100 per mole. So for example, here I'll be using, Mike had pronounced it differently. I call it dabigatran, so I apologize if it's the discrepancy in the pronunciation of this molecule, but this is a commercial anticoagulant that prevents blood clots and strokes. So I'll be using this as an example to describe how we can use iGAL. So if you look at the fragment of this molecule in the red box, I can tell you that it traces back to this aminobenzonitrile up here. So when we want to start our E-factor or PMI waste calculations, we should put this into a commercial catalog. So I have Sigma Aldrich up here, get the dollars per gram quote and do the math to calculate the dollars per mole. And you can see here that we are exceeding our $100 per mole threshold. So at this point, we have to go one step back. And a quick SciFinder search of this will give you many examples on how this compound is accessed through this aminobenzoamide. And if you plug that into the Sigma Aldrich catalog, you can see we are now at less than $100 per mole. And this is what we would call an iGAL aligned starting material. So now you know what starting material you can start with that adheres to the iGAL rule. But most of the time, we do not know the specific procedural details on how that material is manufactured. And this would pose a challenge when trying to calculate our waste contributions towards our total API target. For this purpose, we can use the literature step equation, which multiplies the number of synthetic steps by 37 kgs per kg API. So this is the average waste for literature steps relative to the overall API target. Now this defines and standardizes where we can start our counting for our waste. And this also encourages chemists to think about the synthesis of their starting materials. Our internal process development starting materials can be sometimes complex small molecules in themselves, which most commonly are manufactured externally. And this can result in variability in our industry on how much oversight we have over the raw material synthesis. So if we don't account for this, we're going to be leaving a lot of chemical waste behind the curtain, and most often than not, a lot of cost as well. So at least this gets us thinking about the issues and waste that are coming from our supply chain. Okay, so now I can introduce the iGAL equation. So iGAL is a unifying green chemistry metric that takes into account molecular complexity as free molecular weight, designated FMW, with a fixed target goal for the most innovative and mass efficient process. So iGAL is completely dependent on the free molecular weight of your API. The IQ team found that through statistical analysis of the molecules in the data set, that free molecular weight was a suitable placeholder for molecular complexity. And from the 64 API manufacturing processes in the iGAL dataset, the value of 0.403 was derived as the average waste per unit of free molecular weight. So you multiply your free molecular weight by 0.43, and this gets you your iGAL number, which is your molecule-specific aspirational waste target. So to give an example here with the Bigatran, we know the molecule's free molecular weight. We multiply that by the 0.43 multiplier, and now we get 253 kgs per kg API waste as our aspirational target. So hopefully it comes across that iGAL is a very simple metric that relies on easily measurable data specific to the API target. And this is specific, measurable, and attainable. So if you would like to compare your waste to industry standards, some factors need to be taken into consideration. And one is the consistency of that standard data set, the complexity of your target, and the phase of development of the process you're looking at. So I like to think that if you put all of these requirements as inputs into a reactor, you would get out relative process greenness. So relative process greenness, or RPG, is the iGAL number we just calculated on the last slide for an API process at a specific stage of development, divided by the complete E factor or waste from that process. And it is reported as a percentage value to allow for easier comparison. Each phase of development has different targets, so that a phase 1 or early development process is not compared to a commercial late stage expectation. But the overall commercial goal is of course known, as it is based on free molecular weight. So the RPG matrix is shown here, and it shows how different processes have to perform in different phases of development in order to be compared to others. The phase development ratings are derived from the iGAL dataset via probability plots of RPG at the 90, 70, and 40th percentile. And the rating of excellent, good, and average assigned respectively. So now I want to go into some detail on how RPG can facilitate scoring of the API process and really how to use and understand RPG. So this is completely dependent on the synthetic route of the API. You can see two different strategies here towards dabigatran. The first and second generation route shown here on the left, which in this depiction looks kind of similar to the third generation route. So while the third generation route on the left utilizes a lot of the same starting materials, the late game construction of the target is much different. And we're going to see how that affects RPG in the upcoming slides. So here is the calculation for RPG for the third generation dabigatran route. All we need is our iGAL target waste, which we have calculated on the last slide, as well as the actual observed process waste. And this is the hardest part, right? Calculating your complete E factor and API PMI is a little bit time intensive and takes a lot of document searching. But there's a great tool for this on the ACS GCI website that Mike had alluded to earlier, as well as many cost of goods analyses that integrate this similar calculation. So taking iGAL 253 and our complete E factor that has been calculated for the dabigatran third generation process, 89 kgs per waste per kilogram API. You divide iGAL by your complete E factor times by 100%, gets us 284%, which reaches the excellent category of the RPG matrix, as this is the commercial process for dabigatran. And I can't stress enough how categorizing the development stage makes this metric relevant and time-based. As you can see, the industry average RPGs differ significantly from more of the time-limited development in the early phase through to the more thorough late stage and commercial development. So since iGAL is a metric that provides a fixed waste target for each API based on its free molecular weight alone, the key of upgrading RPG is to reduce waste by improving process efficiency parameters that strongly correlate to waste. In the update to iGAL 2.0, we adopted yield, process yield, and convergence, both commonly used by industrial and academic chemists as key sustainability indicators. So I'll show next how we can get these numbers for yield and convergence and how that affects our RPG. So it is worth noting that molar yield is a greenness metric. It's a measure of step productivity. I definitely do not need to explain how to calculate yield, although we want to keep in mind that yield should be calculated through the longest linear sequence back to an iGAL aligned starting material. But conversely, process convergence is a bit more challenging of a value to quantify. Most synthetic chemists and engineers could look at two different schemes and qualitatively identify which one looks less linear and more convergent than the other. But prior to our work, there was no substantiated quantitative formula for the synthesis of convergence. So we decided to fill this gap. So I'll now explain, how we are deriving this convergence CV equation up here. So over 40 years ago, what I have referenced down here, Hendrickson was one of the few researchers who mathematically characterized convergence in the context of synthesis design. So we're pulling some definitions from his work. In order to calculate your percent convergence of your route, the only extra work you need to do is change your chem draws into color coordinated circles. So like I have here, I've designated starting materials as black, intermediates as gray, and the final API as orange. And now you need to identify these substeps designated SS. So this is the number of chemical steps from a specific starting material to the product. And this will then allow us to calculate the sum of substeps, SSS. So calculated here, 3 steps from starting material 1 to the API, plus 2 steps from starting material 2 to the API, plus 1 step from starting material 3. So then the sum of substeps, SSS, is divided into the number of starting materials, which in this case is 3. This is the substep average, or SS average, and represents the average step distance of all iGAL aligned starting materials to the API, and is therefore a useful proxy for the directness and efficiency of any API process. So where an SS average of 1 would represent the ideal process convergence, we take the inverse of the SS average multiplied by 100% to derive percent convergence. So I would be remiss to not show the new iGAL statistical model now with yield and convergence integrated into the regression. So this diagram can help give you an idea of how the statistical model works. So if we start with a reference process that has 25% calculated convergence in blue and 25% calculated overall longest linear sequence yield. So if we upgrade a given API manufacturing process here, where we upgrade convergence from 25 to 30%, keeping yield constant, we can expect that the average waste or complete E factor will decrease by 14%. In this scenario, if we improve yield by 5%, keeping convergence constant, we can expect an average waste decrease of 9%. And if we optimize both yield and convergence by 5%, you can see the expected decrease in complete E factor is 23% or 63 kgs. So now that I'm showing you the dabigatran first generation and third generation route in a little bit more detail, excluding ChemDraws and substituting in these simplified circles, it's fairly easy to see which one is more convergent. It's fairly obvious here, but now we are able to assign a measurable number to convergency. And through this data, we can showcase the impact of yield and convergence for the process improvements. So here you can see we've decreased the complete E factor. This third generation route has an increase in 21% convergency, an increase to 57% yield, which has now increased the RPG to 284%. And that is the sustainable innovation impact, your change in relative process greenness here, which is quantified as 184%. And now we have our waste reduction, calculated from your complete E factor. So while our API routes are not something we may be readily able to present externally, the route percent convergence, yield, waste may have a little bit more wiggle room. So we have this great way to measure and present the value of our process intensification and route improvements to the broader community and how that impacts waste without showing any proprietary data. Okay, and just like the age-old idiom of comparing apples to oranges, we can now compare apples to apples with the standardization comparability that the SMART iGAL green chemistry metric can provide. So let's see how easy this is. The iGAL Green Scorecard tool is hosted on the ACS GCI Pharma Roundtable website. You will just have to navigate into the tools for innovation, and then you will see the Green Chemistry Innovation Scorecard Calculator or iGAL calculator. So I am going to exit here and go to the website. Okay, so for the sake of time here, I've added the information we need. We can see all we need right now is the project name, the free molecular weight. And now if you want to compare two different process phases of development, we can identify early development here. For the sake of time, I've already inputted the data we need, right? So we need percent yield, percent convergence, and complete E factor, PMI -1, okay? So if we just wanna look at this one route in a vacuum, right? Click the number 1. And down here, you will now see your green chemistry scorecard. So we have the RPG matrix and some definitions up here for reference, but you can see, we can't quantify an innovation impact. We can't see a waste reduction, right? We have nothing to compare to. But at least it's giving you kind of your hard numbers, your iGAL number, and how it performs relative to industry standard. So if 100% RPG isn't as intuitive to many people that are unfamiliar with iGAL, it will then quantify this as 2 times less waste versus the industry average for the specific phase of development, early development. Okay, so and if now we want to compare it to the commercial route, right, we just add in another row here. So commercial, we add our new updated yield, convergence, and our new our waste calculated. And now we can just click 2. Scroll back down to see your new scorecard. And now we can see some comparisons between the different routes. So now we're seeing the innovation impact on the right-hand side quadrant here, which is showing the difference in RPG. And this is, something that a lot of metrics don't provide, is a way for scientists and engineers to understand their innovation impact, right? This is how this green or synthetic chemistry is hard, and at the heart of it is innovation. So this is allowing us to understand how we're improving RPG and relating that back to convergence and yield. And then, of course, we're getting our difference in waste down here, and then your updated, hard stats, your iGAL and your complete E factor, specifying the stage of development, and your new RPG. So this is something, again, where this is colorful. It's easily communicated. And we hope that this can be used in, more higher level, broad communications of waste and presenting process innovation performance. So I will go back to the presentation here. Okay, so future directions and expanding the scope of iGAL. I hope I've demonstrated that the new iGAL 2.0 methodology can serve as a tool to enable significant reduction of global API manufacturing waste. And there are a few high value added extensions we envision for the future of iGAL. So one is to expand the scope of the methodology to include the environmental impact of starting materials for small molecule APIs via cradle to gate, instead of just gate to gate, kind of how Mike was showing for the core production of API. We intend to incorporate a carbon footprint reporting and establish a motivational carbon footprint goal, as this is kind of a more common language in the broader environmental sustainability world. But this will be a challenge, but it will go hand in hand with capturing more of our upstream cradle to gate development. And as pipelines industry-wide are expanding their small molecule assets, kind of literally, to mid-size and hybrid molecules, it would be worthwhile to establish a compound class-specific iGAL waste target. And some current work includes expanding on free molecular weight as a complexity factor by assessing key functional group waste contributors. So to give a little bit more granularity on this, I'll hand it back over to Mike. Did you want to go through some of these slides here?
Yeah, I'll go through these final slides fairly quickly, Sharon.
Perfect.
Thanks for that. So we certainly want to leave a little bit of time here for any questions folks might have, but just wanted to point out to what Sharon already mentioned, that a synthetic oligonucleotide and polypeptide processes are really an area of rapid growth, particularly on the oligonucleotide front. People might be well aware of that now with mRNA vaccines and all that. A lot of those are based off of oligonucleotides. And the interesting thing about these areas right now is that they do all have raw materials or starting materials that do not comply to the iGAL rule, basically. And so that's one interesting concept, is, okay, should they comply to the iGAL rule, or do we need their own metric? And this is just going to be a very important area in the future, or sort of the next generation, where we have to go, because we are seeing more and more products in this direction. And our initial assessments of complete E factor for some of these products and processes are literally can be order of magnitude higher than small molecule. So that's a big challenge ahead of us there. And also these type of assets, also on top of synthetic chemistry, they all will, or most of them will have chromatographic purification, which typically will involve enormous volumes of solvent. So big area in the future, for sure, and it's an area we're excited to be tackling. Next slide, please. And so Sharon did mention the possibility of the carbon footprint, which is often measured as global warming potential. And what that is simply, rather than kg of materials, it's kg of CO2 produced by a specific material. And so there's a very nice paper that was published, the Chem 21 Solvent Guide, which actually did publish the solvent, the carbon footprint for many common solvents. And this is a great asset for people to evaluate their processes with. But notice where you do not want to be here is on the right side. For example, if you look at solvents like THF, acetonitrile, or NMP, they'll all have a carbon footprint of far greater than 5 kilograms of CO2 per kilogram of solvent used. And so that's a very, very significant number. You'd really want to be here over on the far left, where, for example, you have a very low footprint for example, using bioethanol or biomethanol. Obviously, water is great. Use water as much as possible. Next slide, please. And so there was an interesting paper that was published a few years back that did both for 2 routes, a PMI analysis and a GWP comparison. It was real interesting that of these routes, I will say there was a little bit of I'd say a modest PMI increase going from one route to the other, about maybe a 20% improvement. However, when the GWP was evaluated, the carbon footprint reduced dramatically by what we'll call smart solvent selection. So we really think this is kind of going to be an important area in the future, is to start to incorporate GWP. And I think being able to do that, like today for solvents specifically, is right in front of us. Next. Okay, and this is just to kind of wrap this all together. I think Sharon did an absolutely great job of showing, showing the tool that's available on the Farmer Roundtable website, that you literally can go in there and just put the values in for your process. And of course, you can kind of put whatever you want in there, but kind of a key thing is don't worry, your information's not being stored. In fact, you can name the process whatever you want. You can name it Project X, Project Y, and it gives you this exact output. And so, what we're really excited about, as Sharon really drove home, is that we can get an objective measure of our process relative to an average pharmaceutical process. But more importantly, when we start collecting the history, we're able to, on the right side here, really have a metric for the first time that really highlights the innovation, and people should be really that be something that I think is going to show a lot of value, and it's something that's a tool that we can communicate not only to our organizations but even outside our organizations, particularly to satisfy the UN type goals, which are going to be very, very, very important in the future. And I guess the key take home here is really the key focus is, wherever you're at, whatever industry or in academic labs, the focus is to be more sustainable, to be greener, to always be trying to improve from wherever you're at, basically. And I think we'd like to say that, no improvement is, very rarely will improvement not be significant. And that's kind of the key. And we've provided here the tools for you to be able to measure your process greenness in a sustainable manner. Finally, last slide here. I know we're out of time here. We have to acknowledge all the contributors here. Those 13 pharmaceutical groups, companies here working collaboratively. Show the photos for some of the key contributors, and obviously we had 2 universities that contributed, and also the IQ Consortium and ACS Pharmaceutical Roundtables worked collaboratively. And also, we certainly acknowledge the IQ larger organization, which you can read there. It's, as mentioned earlier, it's a not-for-profit organization where all our companies are working together collaboratively to try to advance the kind of the industry as a whole, rather than one company going their own on this.
Thank you, Mike and Sharon, for the concise overview. Great explanations of how iGAL 2.0 works to measure product process greenness based on industry benchmarking. Daring to give a live demo of the scorecard that can be used as an effective communication tool to highlight the innovation impact of our process scientists and showing us what may be next for iGAL. With this, I'd like to open up for Q&A. I think you can type them in the questions box. I don't see question there yet, so let me start out with something. One important topic may be solvent recycling and also reactor and equipment cleaning. Are these included in the waste metrics that we use currently?
Sure, I can take this one, Mike. So these are out of scope for our iGAL waste calculations, and currently, I think in most PMI and E-factor calculations. Of course, these are extremely sustainable practices as manufacturing sites can use solvent recycling. But we are leaving a lot of things out of concept when we don't think about reactor priming and cleaning. But there is a new focus team within the ACS Green Chemistry Institute Roundtable called Manufacturing Mass Intensity that is going after kind of this gap, is understanding what the mass intensity is in the manufacturing setting. And I think that will be a useful tool or metric that will come out of the roundtable, and hopefully we can try and see how we can integrate that into iGAL in the future.
Great, thank you. Anybody has a question? Okay, there's one. Let me read it. iGAL is a great tool to evaluate our process. Is it possible we can create our individual account to save the data for future comparison?
I guess the answer to that is I'm not sure of the answer to that of the answer to that. I know that people typically will use the tool and perhaps print it out, but I'm not sure if that can be saved as a local file. Do you know that answer, Frank?
No, right now you can only save the picture by right-clicking on it and save as, but there's no database to save the data at this time.
And so I think that sounds like that's a good idea for a future refinement, to make that easy and perhaps port it out into Excel or something.
Yes. Okay, next question. How difficult is it to change, oops, sorry. Is it to change a process once it has been FDA approved?
I could take that on. That is not easy to do, because a post-approval or a type process change has to. People think of it, oh, it's just one, changing in one regulatory geography, but the reality is, for global processes, you're filing in the U.S., EU, Japan, and then basically a ton of other individual companies all have different regulatory review processes. So it could take literally, if you have a process change you're going to make, a significant one, it could take in theory up to 3 years to get that to stick in all geographies, and you have to have flavor management while that's occurring. So there's quite a bit of incentive to get the green piece, the sustainable piece, done at the time of the initial filing. Although that option's available, it's just more difficult present day. And I think that's one of the things that would be, I think, very important would be, we'd like to see some regulatory relief on perhaps accelerated, expedited reviews in the future for making sustainability improvements. I think that would be a great objective to have.
I think that was actually one of some of the initial things that the IQ Green Chemistry Working Group participated in was, conversations with the FDA on how to reduce those regulatory barriers for refiling with a sustainability, driven purpose to that. Hopefully we'll come out with a few more items and initiatives on that topic.
Great, thank you. Next question, how will you be estimating the GWP, so the global warming potential, the carbon footprint? Is the GWP linearly correlated with PMI?
I could take that one on. The latter part is linearly correlated with PMI. No, that's not totally clear. That's something we'd have to build out and evaluate it. Right now, what we do have is good, reliable data for solvents. And you'll note there were some solvents missing there, but we do have tools. Some are proprietary tools, but tools we could get access to that calculate that. For actual process materials, it's much more difficult, and so that's something we just have to work on. I think in the near term, I think the solvent piece is probably going to be the biggie.
Yes, and there's of course two parts to the GWP. There's the production part, so what's the GWP of your production process, and then there's the part of the GWP that's associated with your raw materials. That's why Sharon mentioned it's so difficult. All right. Next question. Do most reagents, solvents, catalysts, ligands also conform to the iGAL $100 per mole rule? What about transition metal catalysts? Manufacturing them requires significant resources, which would not be captured in a typical waste metric. Good question.
Yeah, I can try and take this, and Mike, you can add on if you would like. Yeah, I think this almost, when you think about transition metal catalysts, right? You almost need that more life cycle analysis of that raw material to fully capture, the waste impact of that. And it becomes a little bit more esoteric on using the literature steps rule, right? How many steps does it take to make palladium on carbon, for instance, right? That refinement. I think it's definitely a challenge. I think if we could do our best to adhere to the iGAL rule, especially with complex ligands, right? That they bring their own waste to the table, especially. But yeah, that is a challenge we have right now until we can really start integrating the life cycle analysis and impact of some of these raw materials.
Great, thank you. Next question, interesting one. Often API quality drives the route, which often correlates with yield. But any thought of how that metric, so quality metric, I guess, could be integrated into the tool?
I think on that one, that's a great suggestion, and certainly potentially adding a component down the road for purity is important, because certainly, as people know in this field, that 99% purity or greater is very different than 98 versus 97 versus 96, and that'll be even more important as we move towards the larger molecules, basically, because there will be very different grades of purity to address there.
Thank you. Okay, a practical question. Can you provide a time estimate, time commitment for these calculations from start to finish?
Yeah, I guess, well, you could imagine if you have all the numbers that you need, calculating GAL is quite quick. But with that said, I would say the average time, if you're using the PMI calculator on the ACS website, and you have all of your batch records in hand and your waste and your inputs in hand, perhaps a couple hours to calculate the E factor of a process. This could potentially be improved with more advanced software. And I would say the convergency, if the full start to finish of your route, wouldn't take more than I would say 10 to 15 minutes to get that convergency number. So it is, I think the hardest part is the waste calculating, right? That is the most time consuming. But if that becomes a standard practice in your organization, hopefully it becomes less of a burden and more automated and digitized.
Thank you. And we'll do one more question. Why does CEF now include water as opposed to the original definition of the E factor? Is this water used in actual chemistry or for rinses, cleaning?
As I think Sharon mentioned, the water is actually a big part of the chemistry, actually, because there are quite a bit. A lot of times it can be maybe less frequently in the actual chemistry steps, but workups use extensive amounts of aqueous, and the important part is we make sure we capture the full process, which includes any type of workup, which is very frequently aqueous. And we found that basically is a very significant component of the overall waste profile. So important to capture, and in certain geographies, actually aqueous waste is very, very significant to deal with and remediate.
Thank you very much. So this answered all the questions. Thanks again for the presenters. Thank you for your time to prepare these excellent slides. Thanks for the audience and your interest to attending. And with this, we'll close and wish everybody a nice weekend.
Thanks all. Good day.